Welcome to a whole new paradigm shift, or rather, welcome to the very beginning of a possibly exciting, frightening paradigm shift that's so new, we're only going to be beginning to figure out how to talk about it here tonight with the three experts we have. Are we about to head into a brave new and frightening world in which we may seriously consider suing our grandparents or even our great-grandparents because they didn't take care of their genome and we're having a little trouble about it? Are we possibly heading into a world in which we can take a pill to change our feelings about sexual orientation or about violence or about war? Are we not already in a world in which epigenomic effects described by scientists are already the subject of law cases? There's a lot of legal implications in what we're about to hear. But I've been warned by a number of people, including Dr. James Watson himself, Dr. Eric Kandel, and some of the older men and women who are giants on whose shoulders we stand, to be very cautious about what we talk about here tonight, because it's being overhyped, they say. And yet I have yet to talk to one scientist in epigenomics who begins by saying, we've got to be careful now, and within 5 minutes isn't ecstatic, nervously ecstatic, about what this all may portend, about what we will learn about who we are, what we are, how evolution really works in ways that turn out not to contradict Darwin or Lamarck, and which may mean even that we may be beginning to enter an era in which we might ourselves now, let's hope cautiously, take over the control of evolution. These are no longer ridiculous questions. That's the point of what we're going to try to make a little sense of out of tonight when we talk to our experts. So first, let me introduce our three experts one by one. Our first participant, Randy Jertle, was nominated as a Time Magazine Person of the Year for his pioneering work in epigenetics. He headed the Epigenetics and Imprinting Laboratory at Duke University until last year. Now he's a visiting professor at McArdle Laboratory for Cancer Research at the University of Wisconsin. Welcome, Randy Jertle. Also joining us is Frances Champagne, an associate professor in the Department of Psychology at Columbia University. She researches the role of epigenetic mechanisms when it comes to environments and gene activity. Frances Champagne. And our final participant, Dr. Jean-Pierre Issa, is a professor of medicine at Temple University and the director of the Fels Institute for Cancer Research and Molecular Biology. He's working on the large-scale mapping of epigenomic patterns in normal cells, in aging cells, and in cancer cells. Jean-Pierre Issa. And now, since we have an enormous task ahead of us, we thought we'd start with a 60-second teaser, just to give you a sense of some of the work that is nailing down these possibilities I was just mentioning.
In the 1980s, when Dr. Lars Ola Bigren investigated the records of a remote Swedish village, births and deaths, feasts and famines, the data revealed something strange. Experiences or habits of the fathers appeared to be somehow physically inherited by the sons and grandsons and great-grandsons. Scientists became detectives and opened up other cases. The data from the Dutch Winter of Hunger and the Chinese famine in the wake of the Great Leap Forward revealed similar mystifying patterns. What was happening? Was Jean-Baptiste Lamarck actually onto something when he said that a giraffe that really stretches its neck to reach even higher leaves would give birth to a longer-necked baby? Can experience change our hardwired DNA and our genetic destiny?
That's the old paradigm that we were all pretty well locked into up until about 10 years ago. We have hardwired DNA. It's changed slowly by cosmic radiation, perhaps zapping it, so every now and then there's a little bit of variation. But now, epigenetics is running through conversations all over the planet. Jean-Pierre, let me start with you for a simple definition of epigenetics.
Well, when people ask me about epigenetics, Bill, I just tell them, look at me, or look at the mirror. What do you see? And what you see if you look at me, you see skin, you see eyes, you see hair. If Superman was in the audience, you'd also see liver and lungs and bones and all of that. And we have more than 200 different tissues in our bodies. They're very different, they don't look alike, they're very stable. And here's the mystery. We have 200 tissues, one DNA sequence. So Dr. Watson, who was speaking earlier today, when he discovered the structure of DNA, couldn't explain that. How could you go from one DNA sequence to 200 different tissues at the same time in the same person? And so in the 1950s, Conrad Waddington, a biologist, was studying this question and postulated there must be something that works on top of the DNA sequence, and this is where the term epigenetics comes from, epigenetics. And at the time, he didn't know what it was, but he said there must be something that modulates the genes in different tissues. We now know what it is, and that's what we're going to be talking about today.
We now have an idea about what it is. You have it all 100% nailed down?
No. I wish we did, but we have some idea of the mechanisms of how it is possible to have 200 tissues with a single DNA sequence.
So, epigenetics. Francis, a little bit more on the word epigenetics. What does epi mean?
Epi and top of, above. Genetics, obviously, now we refer to that as the DNA. So these are all factors that come around the DNA and ultimately affect how likely it will be that the DNA is read or not.
Something like the old nature-nurture debate, this being the nurture of the environmental part, these epigenetics, as we'll explore, may be affected by experience, by trauma, by the environment.
Exactly, and when you think of nurture and nature, really what epigenetics represents is the interface between those two influences, how our experiences come to modulate what genes are turned on and what genes are turned off.
We've gotten the impression over the last 10 years that the old nature-nurture debate, 60-40, 40-60, 70-30, 30-70, is really 100% of each. It's always both, and it's not even the right question.
Always both. Wrong question.
Exactly. Randy, hot field? I think we may have a graph that may point out what you're about to tell us about the excitement you all are experiencing who are at the center of the discoveries you've been making.
Yeah, it's a very hot field. I mean, there's a lot of people now working on this field of research that weren't working on it before, a lot more publications, which you can see in this graph. For those of you that go into a cold sweat when you see a graph, this is not very difficult. This is number of papers that is plotted against years starting in 1990. And the important points that you can take from this graph are really two. If you look at it and analyze it, you can see that this is an exponential curve. It's growing, the field is growing exponentially, and it's doubling every 2 to 3 years. By plotting it this way and you focus on that little asterisk, that's where there's an inflection point between, in effect, if you were watching this from above, it would look initially from 1990 to about 2003, 2004, that virtually nothing was going on in this field of research. But after 2005, it's gone vertical, or as they would say now in the vernacular of the times, it's gone viral. And this is what we're going to be talking about today. I mean, frankly, none of us would probably be on this stage if it wasn't for the fact that it has gone viral, and none of you would be probably in this audience.
And just for a moment to consider the implications, which we'll get into later, of all of this, what we saw from the village of Overkalix, passed down through generations, possibilities of trauma or famine affecting what happens in people's physical lives 3, maybe even 4 or 6 generations later, children of PTSD in various horrible things that have happened in the past bearing these marks. Enormous implications for law, for society, is it not?
Yeah, enormous implications. And one of the most interesting implications is that about 10 years ago, when the human genome was sequenced, some scientists were saying, well, that's the end. We're going to understand every disease. We're going to understand every behavior. And it turns out we didn't, because the sequence of the DNA isn't enough to explain behavior. It isn't enough to explain diseases. And that's where I think the field of epigenetics took off, because even hardcore geneticists started saying, wait a minute, why are these people with the same DNA behaving differently or having different diseases? And they started paying attention to us.
On that point, people with the same DNA, we all have heard of identical twins, relatively young ladies here, but the same DNA at the beginning, and as they grew to be older and older women, differences appeared. What is this teaching us, Francis?
Well, when the first discovery came to light that we have these identical genomes, but they start diverging in terms of their character, in terms of what they're doing in the individual, there was evidence that there was an epigenetic divergence. So when you're young and you're twins, you have very similar epigenomes. When you're older and you're twins, you have very divergent epigenetic features. And so the implications was that as you walk through life, as you have your unique experiences through that lifespan, your epigenetic changes are mirroring those experiences
So to be specific here, what you all have learned is that genes can turn on and off and that they need instructions. They don't just sit there and do what they do. They need different instructions and may get different instructions to do different things at different times in a person's life.
Exactly. Having DNA is great, we need it, but we need to also know what and when genes need to be turned on and turned off. So the light switch, the timing is incredibly important to development and functioning.
Well, to anchor this in something physical, let's spend some 10 minutes here talking about the mechanics of it, at least what you all know so far, because I get the idea you all feel you're just at the beginning of it. How epigenetics works. We have a drawing that we've put together here that I'll ask each of you to help us explain. And it's a little bit unfinished on purpose because the impression I get from you all and your colleagues is that you're just at the beginning of something very exciting. So let me first ask you, Randy, over there on the right, this is obviously the double helix of the basic backbone of the DNA that we're all so familiar with, and that Dr. James Watson was, along with Francis Crick, discovered some years ago. But over on the right, what does that ME stand for? Me?
No, it stands for methyl group, and a methyl group is a carbon atom with 3 hydrogens attached to it. That methyl group then with proteins can be either put directly onto the DNA, or it can be added to the spool that the DNA is wrapped around, which is comprised of histones. And in the process of doing this, when those methyl groups are put onto those 2 regions, one in the histones and one in the DNA, it will tend to cause that DNA to condense, and therefore it is inactive and cannot be transcribed or make proteins from that gene. Whereas over on the other side, you have the AC, which is an acetyl group that can be put on histones, and when that occurs and the methyl groups are removed, the DNA opens up. They're still wrapped around those spools, but as a consequence now, factors can come in and cause that gene to be transcribed and you get protein.
A question before we get too deep into the jargon there. These things can be put on. It sounds like you're talking about things that environment might cause to happen or experience might cause to happen.
That definitely does happen, and when they're put on, you can also put them on appropriately in normal situation, or the environment can alter which marks are placed, and you can cause abnormal situations.
Okay, so that's part of what the epigenome is. Now, right. Jean-Pierre.
But just to be clear, they are put on, for the most part, based on our DNA sequence, and this is essentially what happens in embryogenesis.
They aren't or are?
They are. Now, so most of the programs is determined. However, the program is not 100% accurate or efficient.
One is right after fertilization in the embryo. The second is right after birth, where you have a lot of remodeling, particularly occurring in the brain, which is what Francis is going to talk about. And later on in life, the wobbles can occur because things don't work quite as well, which potentially can give rise to cancer, which was what Dr. Issa is going to be talking about primarily today.
Right. And while we're at terminologies here, what about histones? Jean-Pierre, what are histones?
So histones are these proteins that form the structure in which DNA is located inside the cell. There are these round structures that you see on this drawing around which DNA is wrapped. And histones are essentially the gatekeepers for DNA. They can either hug the DNA and hide it away inside the cell in a drawer so that the DNA is not accessible, it is not made into proteins, or they can open it up and say, here it is, make RNA, make proteins. And that's the central difference between, say, a muscle cell and a liver cell, in that in a muscle cell, there are histones that are open up and say, here, make all the genes you need, make all the proteins you need to become a muscle cell. And in the other cells, these proteins are hidden away in a compartment by these histones.
So the histones are sort of huggers, and they can turn off a gene.
That's right.
The methylations are tags that can turn off or on a gene, as the case with the histones. So this is some of what you all have discovered about the mechanics of the physical epigenome. And they may be, the point here is that they may be affected, whether they're going to hug or not hug, turn off or turn on. They may be affected by the environment and things that happen to the person.
Exactly. So we have a very stable genome in blue ink, but we have these pencil drawings of epigenetic changes around that blue ink. And certainly you can take a methyl group and you can add it to DNA, you can take that same methyl group off. You can change those histone proteins very dynamically. So even learning something new will induce an epigenetic change. So these are very dynamic processes.
So you all are learning how nature works, and I can see the humans in you and the people you're talking to say, if nature can do it, we might be able to control it a little bit. And pencil up here is, in other words, sort of like software.
Exactly.
And the blue pen drawing is what we've all got as hardware. And the software can turn on and off. I'm reminded how often, when we have trouble and have to call up the IT departments, how often it's mostly the software, not the hardware, that goes wrong.
Right.
Much more often. And so you scientists have seen a real potential to move in here at something very exciting. Triggered by outside events. Now, there's one specific question that I have here. This is what we all learn in middle school, is what gets passed down from generation to generation, the DNA part, the part in blue. And it goes into the eggs and sperm of the offspring. And it splits, and the offspring get half of their fathers and half from their mother, and that goes on down. My question, looking at this, is whether these first three, methylation, acetylation, and histones, which are also physically attached to the genome, do they also split and get passed down through the germline to the moment of conception? Do they become part, or do they get stripped off?
Well, the historic view was that they are stripped off, and they are to a large degree.
At the moment of conception
stripped
for the
off.
next...
At the moment of conception, histone proteins, for the most part, aren't in sperm. There's a different protein in sperm. And when the two genomes come together to create the offspring, all of these epigenetic changes are supposed to be stripped away. But I guess the more recent and exciting and kind of mind-boggling finding of late is that perhaps some of these changes aren't stripped away and that they are carried forward into the next generation.
So it's not just the DNA pairs that go down into the eggs and sperm of the next generation.
That's right. It's carrying along methylation baggage and histone baggage.
Right along, into the moment of conception.
Yes.
That means things that were affected by the environment of person A, in the child of person A, they may actually go along.
They may. I mean, this is the area of speculation that we
So
really
we have to be
need
cautious
explore.
here.
We have to be
But
cautious.
you have a lot of reason, I see a smile on your face, Randy, to suspect it may well be true.
Well, it's true at least in one model system that we know of, which is the Goody mouse system that we're going to be talking about.
Right. But I've also heard all of you and others say that we already know that this happens in plants and insects. Is that true? That epigenetics get passed down without being stripped off? Jean-Pierre?
It is true. Epigenetics plays actually an evolutionary role in plants, in that there is some epigenetic wobbliness or epigenetic variability in plants, such that when plants are exposed to the environment, say severe drought, some of them will survive thanks to very specific epigenetic changes that they have in genes that allow them to survive drought. And then these plants will transmit that information to their progeny. But it is wobbly. After a few generations, it goes back to baseline. So this, as opposed to the DNA sequence, which is fixed, DNA sequence is a binary switch. You're either on or off. Epigenetics provides you more of a variability, and then that is an evolutionary advantage for periods of drought or...
But Jean-Pierre, would it go off if, for example, they were continually in a stressed environment? Let's say, for example, increased salt content or something. Probably not.
Right.
It'd be selected for.
So it's only if the environment goes
back to normal.
To normal, where the drought disappears, then it sort of gradually over
Goes
generations goes back to baseline.
4 to 5 generations, probably gone.
Well, this term wobbly, which doesn't sound overly scientific, is intriguing. I guess you're talking about something that provides a further variation of possible varieties that may be there in the next generation to meet whatever challenges come down the pike. A little bend for the reed in the wind rather than having to break.
Exactly. We wouldn't be here, we wouldn't be humans without variation. Evolution requires variation in order to work. We have thought for many years that evolution works only on genetic variation, but now we understand that it can also work on epigenetic variation. The DNA sequence isn't everything that matters for evolution. And so the ability to generate this variation is actually important for evolution. And we probably are here thanks in part to this variation.
And us wild and crazy humans who seem so much more destructive and productive than any other creature wouldn't be here without this as well.
Well, that may well be the case. That's actually the work maybe of Francis, but I work, more on cancer. But yes, it's true. And I certainly, from my field, I could tell you that that is central to many cancers, and that cancer is inevitable because of this variation.
We'll get to that in a minute.
Yes.
But just to make sure that we don't pass up the implications already, you guys are finding mechanisms by which events in a person's life, like in the overcalyx famine records we see, may be passed on to grandchildren, even to great-grandchildren. Possibly a permanent change in the genome. So we have a kind of responsibility for taking care of our genome so that our great-grandchildren won't sue us? Is that a possible question now?
Well, whether they can sue you or not is quite another question.
If you're still around.
But it's true. I mean, there is more and more evidence that grandparental, even great-grandparental environmental exposures can impact you. And so these are big questions.
And there have been fascinating studies by the adult children of Holocaust survivors, by the adult children of survivors of the Cultural Revolution, the adult children, well, in fact, the children who were in their mother's wombs at the time of 9/11 having higher cortisol levels, the stress hormone, which itself can possibly, as I understand it from the research, leave scars on DNA as well, and so stress itself. So, first implication here, we may be responsible to try to keep ourselves as relatively stress-free in our own life as possible so that our great-grandchildren won't be overly stressed either, long after we're gone. You're laughing, but that's because you guys are opening up the possibility of this being a sensible question. All right.
That is an essential point, but keep in mind that there are also periods of time in our lives where we're more susceptible to that. And it's really the mothers when they are pregnant, the fathers when they are conceiving. And so there are periods of time where we have to be much more careful about our genome than perhaps other periods of time.
So the more we learn about which periods of time to be more careful,
That's right.
the more knowledge and responsibility, but potential also for good and preventing suffering will come along.
Yes.
So let's look at 3 different breakthrough studies before we get too wild in our speculations here. So we really get a feeling for the hard work that you all and your colleagues in the scientific world have done. Let's start with a study of mice. And Randy, I understand these are known as agouti mice. Is this something from the high Andes, the agouti mice? What does agouti mean, and why is this study that you did now so often cited as being important for understanding epigenomics?
Well, agouti just means brown.
Ah.
It's not too exciting. So you have 2 mice. I call these the agouti sisters. So from the hot Dutch study and the Chinese famine studies, you knew that something occurring very early in development, and literally in the first trimester of development, ultimately gave rise to increased susceptibilities to diseases, including cardiovascular disease, obesity, diabetes, and a doubling or tripling of the incidence of schizophrenia. So severe lack of nutrition early in development results in all of these disease characteristics 20, 30 years later. The real problem, though, was what is the mechanism by which the glue, the gravity, the molecular mechanism that holds these 2 disparate time points together. And this is how why this study is so important. So the only difference between the brown mouse and the yellow mouse on this slide is what the mother was exposed to when they were in utero. These are genetically identical mice. The mother on the left, the yellow mouse, that mother was just eating normal mouse chow. The mother on the left was eating mouse chow that was supplemented with things like folic acid, choline, etc. Why those compounds? Because they're the ones that provide the methyl groups that we were talking about before that are placed down onto the DNA. All those methyl groups that are needed for programming come in from our diet. So that's why this early time point is so important. So here you have a mouse that is small, lean and clean, doesn't get diabetes, doesn't become obese. The only reason is because what had happened and what that offspring's mother ate very early in development. And what we showed is in this system anyway, that molecular mechanism that relates these 2 time points together is DNA methylation at a region that's upstream of the agouti mouse. So for the first time we showed that small changes in nutrition can alter disease susceptibility, not through alterations in the genome, but by alterations in the epigenome. And this is fundamental.
Because it's epigenomic.
It's
To be
epigenomic.
clear, it's in the parents of these mice.
The parents, the
When
mother.
they were in utero, when they had already been conceived.
The mother was eating a nutritionally supplemented diet. The brown animal, for example, was supplemented, and the one on the left-hand side, the yellow animal's mother was just eating normal diet. So the only difference is what the mother was eating when they were in
You gave the
utero.
mother of the orange one more vitamins.
Less vitamins.
Less vitamins.
Right.
And that meant that certain preventive genes weren't turned on or off?
Off.
Weren't turned off.
Off.
The one on the right had them turned off. and you traced the mechanism of it.
It's DNA methylation. We know absolutely that's the case in this model system.
So,
And you have obesity. The yellow animal gets a higher incidence of cancer and has a higher incidence of diabetes. So by supplementing the mother's diet early in development, you prevent all of these chronic disorders.
and one can already imagine the potential for, as I believe are already in some law courts, legal responsibility of mothers, or legal responsibility for employers in the environment of fertile or pregnant women, or just fertile women.
It's possible.
These
I
are-
don't get into the legal aspects. I have enough travel with the scientific
Right. But the work you all have done has now made this a very sensible question to ask.
Exactly.
And there's real potential for good from this if we can understand how to protect.
I like to think about and focus on the good aspects of this rather than the negative aspects.
Right. And again, the question, whether these changes could be permanent going down through the offspring of these mice and their offspring.
In this model system, if this methylation change that we see upstream of the agouti gene, this can be passed on from generation to generation through the mother. Through the father, that is not the case. So this ability to have transgenerational inheritance of epigenetic effects appears to be dependent upon the gene that you're looking at. It might even be dependent upon how it's passed down through the mother or the father, that type of thing. These things have not been worked out well, even in this model system, let alone in humans.
But it's beginning to be nailed down.
It's
You
beginning
guys
to be
have
nailed
published in
down.
peer-reviewed journals.
So here you have a video of these little agouti mouse running around. Again, these are genetically identical animals, and I think you can clearly see the yellow animals are massively morbidly obese compared to the brown ones. And again, that difference is completely determined by epigenetics, not genetics.
Now, the question has to be raised here. These are mice, but these are mice, not humans.
These are mice.
You have good reason, though, to believe that these may be mechanisms that may be not totally unlike our own.
It looks like from some of the model genes that we've been looking at, for example, these imprinted genes, which we might talk about later on, that indeed is the case. You can see environmental alterations, the epigenome of these regulatory elements that control the expression of these genes.
So Francis and Jean-Pierre, when you learned about what Randy had figured out here, it was of interest?
It was a great interest, and what's wonderful about this model is you can see it, right? We've got a yellow obese mouse and we've got a brown mouse, and they are genetically identical, but clearly not the same.
For epigenetic reasons that you can identify.
Exactly. Yeah. So I think it's really strong evidence, and it's just so visually effective in explaining epigenetics.
Well,
And I say it's not just visual, but this really was one of the first times that the relevance to disease was very easy to show, because for many years we've been studying epigenetics as a mechanism of differentiation, lung versus liver versus blood. In here, what you see are mice that have exactly the same genome, yet have very different phenotypes. They look different, and phenotypes are diseases. Diseases are phenotypes. So you can think in this case of, well, they have a different coat color, but if you could see it, you would also see different susceptibility to cancer, as Randy mentioned, diabetes, obesity, potentially other diseases, neurologic diseases, all of those linked to differences in DNA methylation, not differences in DNA sequence. So this was a very strong indication to doctors out there that epigenetics could be relevant to disease, not just to differentiation.
So for every research area, there is a model system that's optimal. And for the field of epigenetics, this was the optimal system for looking at this initially.
breakthrough.
Breakthrough.
Let's go to another breakthrough study. Francis, moving up, or is it over from mice to rats? Or down? Well, we're not going to get into that. Rodent politics we don't need any more of that.
Very complex.
Francis, you worked with rat pups, I believe. We
That's
have
right.
a picture of also the general field of maternal care in various species, which is what's really important here, whether it be in humans or in primates or monkeys or in these lovely little rats in the upper right-hand corner. We've all known intuitively down the eons, no doubt, that maternal care can make a big difference, but you began to zero in on it with your study with rats and they're called rat pups, the little kids, I think.
The little pups.
So what did you discover?
So, we were really interested in this question of how maternal care could shape offspring development, something that we intuitively understand. Our experiences growing up, particularly with our parents, would have this astonishing effect on the way we function. And so we moved that into the lab so that we could really nail down the mechanism. What is it that mothers are doing to their offspring to change their development? And we were particularly interested in how they respond to stress, the offspring. So how we respond to stress is really a fundamental aspect of how we interact with the world. We might be more stressed, we might be less stressed. And certainly in our pretty genetically identical animals within the lab, there are very big differences in stress response, with some showing very high stress reactivity and some showing low. And when we looked at the amount of maternal care those high and low stress animals were receiving, we realized that the individuals that were very highly reactive to stressors were not getting as much maternal care from their mothers during the very, very early stages of development. And rats, unlike humans and primates, spend a lot of time licking their babies, so it's really all about the licking for when you're a rat pup. And so how much licking you get from your mother, which is really a critical form of tactile stimulation between mothers and infants, would predict all of these long-term outcomes: stress, cognition, and a number of other effects on social behavior, for example. So finding this, we thought, okay, this is great. We know the mothers are shaping these systems. How are they shaping them? So if we look at gene activity in the brain of adult animals who had received high or low levels of licking, we found that the individuals who had very low levels of licking when they were infants, when they were adults, certain genes that help you respond to stress and kind of recover from stress were shut off epigenetically. So we looked at genes such as the glucocorticoid receptor. We found that if you had high levels of maternal care, this gene was fairly active and expressed. And what that meant is those animals could get stressed, but they could recover from the stress very rapidly. Those individuals who did not get very much licking and grooming had very low levels of these receptors because they were epigenetically shut off with DNA methylation. So there's lots of methyl groups attached to the regulatory region of the glucocorticoid receptor gene. Shut that gene off, and these were going to be very highly reactive to stressors. Now, when we looked at the female offspring of mothers who engaged in high or low levels of maternal care, we saw that the females inherited the variation in maternal care that they received. So if they got lots of licking, they also engaged in a high level of licking towards their offspring, and their offspring also engaged in high levels of licking, grooming towards their offspring. So licking, grooming, this maternal behavior that is shaping epigenetic profiles in the brain, affecting the way individuals respond to stress, was also being passed down generations through the female line. And what we determined is that the more licking and grooming you get during the first week of life, the more active the genes are in your brain that regulate maternal care itself. So maternal care tends to turn on genes such as the estrogen receptor, which makes you more sensitive to the hormones that females have during pregnancy that prime you to be maternal towards your offspring.
So I'm beginning to hear something that sounds like it may have an enormous amount of new implications in what have been controversial subjects in the past. You have what, if I can borrow a term from Bruno Bettelheim, a cold mother doesn't do much licking, and a warm, nurturing mother does a lot. And of course, he was famous for having postulated a long time ago that cold mother syndrome led to autism.
Right.
Now, you're suggesting here that cold mother syndrome, in rats at least, might also lead to serious problems.
Well, low levels of maternal care, and these are very stable traits in these females, has all of these long-term and perhaps even transgenerational consequences. But when you look at why they're engaging in low levels of licking, grooming, then you get a very interesting answer. So it's really the environment writ large. So
I want to be very careful about what I just said, because that idea by Bruno Bettelheim gave a lot of guilt to mothers of autistic kids that has since been overridden, and the understanding is now is far more complex than that, and autism may not be anywhere near as simple as that. But this is just to give a sense to our audience that what you all are opening up again is a field of questions which can have very controversial edges to them, and it's very powerful.
Right. And there's a whole field of medicine now that is looking at fetal origin of adult disease. And so this is a complex term, but it's a very simple idea. It's the idea that what happens in the womb and what happens very early on in life might influence your disease state or maybe your psychology or maybe your rate of depression 50 years later or 40 years later. And that's actually pretty amazing from a medical terms, because when a patient comes and asks, why did this happen to me? You know, you go back a few years and you start thinking, did they smoke? What were their exposures? You don't think to ask what happened when you were born, or what did your mother eat when she was pregnant with you. Yet that could be relevant to the diseases that people get older, at later ages.
I want to take us in a little bit of dangerous territory here. I'm thinking about the Civil War. I'm thinking about the American Indians and others who have suffered enormous. You were going to.
I was just going to say that the work that Francis has done is actually has really turned the field, I think, of psychology on its head. And I have an interesting little tiny story about this. In Wisconsin, there's a person that I know, and he said, and he's in the psychology building. I said, you have a research lab in the psychology building? He said, yeah. And he said, you know what? They had to actually expand all the sizes of the doors. I said, why is that? Because he said, I couldn't get in my lab equipment. So research now in a psychology building is now becoming wet lab research. And the reason for this is because of the work that you and your colleagues have done. There's no doubt that it has been changed forever.
Well, this is what I was saying at the beginning of a new paradigm, quite possibly, although all kinds of experts are very nervously ecstatic about it, but to introduce one of the reasons why. What you're just suggesting here is that it may be that trauma, if we could take low licking and expand the idea, leaves scars on the DNA one way or the other. You're nodding.
Yeah, exactly. And going beyond just licking and grooming, there's animal models of abuse, and certainly in primates, there's evidence that abuse leaves this lasting epigenetic change, even in the human brain.
Wow. Now, at that point, let me just expand a little bit to something else. War, horrible genocides have been visited on all kinds of groups of people. They've been discovered in primates by Jane Goodall and other scientists. They seem to be something that cruelty seems to be selected for in evolution. As Edward O. Wilson has once said, the thing about us humans is we're just so damned dominant, because sometimes we're dominant by one group being more cruel than another and hurting another group. This leads, if I'm not going too far, to the possibility that cruelty may also be selected for because it can make a group more dominant, because by causing trauma to another group, it diminishes the powers of that other group. For a while, they have more cortisol. Is that a reasonable question now?
No question's unreasonable. but what I think, certainly, I mean, you could have all of these factors that are contributing to higher levels of aggression in an individual, and there's certainly that could have an epigenetic basis. But at some point, you have to give way to intergroup relationships. So it's not enough just to be aggressive to everyone around you. If you want to live and survive and prosper, you have to get on with your own group.
Well, the idea lately is that there's, within groups, altruism is selected for so that your group can be strong, but between groups, the group that can be most cruel to the next group is going to be dominant.
But I think the point is that there's no selection for one thing. There's no selection for cruelty. There is selection for variability. Because if you take a group, under some circumstances, there might be war, and under those circumstances, groups that have more aggressive traits might survive. But there might be other circumstances where there is no reason for war. It is those groups that, for example, are more intellectually inclined, who can discover agriculture or can discover ways to grow food, that are going to be selected for. So ultimately, evolution doesn't like one thing. It likes to have a diversity of things in a given group so that under the right conditions, that group would survive no matter what. And that's where epigenetics also provides this flexibility. So you can imagine where you have an extended family where, a couple of the uncles are very aggressive and a couple of the aunts are very nice and nurturing, and that the group will survive no matter what the environment is. And that's what matters, I think, to evolution.
And a note of caution against myself and all of us here. I just made some leaps there, but the implications of what you're finding about what passes on and weakens later generations from lack of caring may have implications for how one group of humans is kind or not to another group of humans, which points us towards thinking about our desire for a peaceful world in which all the groups have to try to not traumatize each other too much.
Yeah, if trauma has these intergenerational effects, and certainly the evidence suggests that it does. And then I think we get back to this wobble. So we may need this aggression or hostility to our other groups to last a few generations, but we may not want it to last forever within our genes. And so that's where the wobble, this unscientific wobble, may be very handy to get rid of these effects after they've been incorporated.
This gets back to the whole thing about the genome being really quite static, but the epigenome being plastic. And that's the advantage of having these two systems. You've got something that's more or less rigid and can be passed forward from generation to generation without much change. And then you've got this plastic situation that can respond rapidly to the environment. And that gives rise to these wobbles, you know, as we were talking about the wobbles. And that can either be selected for or against in a short term, or if the environment stays this way, potentially in a long term, and it will be fixed at that point.
And what's getting exciting about what you all are saying here is that we humans in this auditorium, and you guys who are doing the hard work, now understand this. So we may have the potential.
We understand the mechanisms by which it happens,
right,
but we don't know right now the genes that are involved in this, particularly in humans, and the regulatory elements that are involved in these processes.
Right, but
this
we're
is
learning.
what we need to know now.
But we're learning,
We're
so
learning.
it's opening the possibility that we may be able, as one movie director who had a lot of movies that showed the bad side of human nature, and he was an interviewer complained about this, and he said, yes, but it shows we can get our mind around it. You're showing us we can get our mind around this bad side of human nature, so we may be able to choose to be kinder so that we don't perpetuate the cycle of violence down through the generations.
So Bill, we're scientists. The important thing today is to go beyond speculation. And the real nice advances are that we can measure these things. Really what has made the difference in the past 15, 20 years is not the understanding of epigenetics, since it was postulated in the 1950s, but now we can see it. We can measure it in the lab. These tags that you talked about, these bookmarks, you know, the methylation, the acetylation, you know, we could actually take your genome or take somebody, you know, in this room's genome and study the methylation states and look at all the gene and ask how much methylation is there in this gene, and does that correlate with, say, aggressivity? Does that correlate with having diabetes? And so we are asking questions, and these questions need to be answered with data. And we're all here interested in actually doing these studies rather than simply speculating about this. But we're excited because the implications are enormous.
Right. Well, now let's go to a third breakthrough with some of your work, Jean-Pierre. You have taken this, to come back down to earth, to an actual work in cancer.
Right.
And you've had some remarkable successes with it.
Right. What you see here on the left is a CAT scan of a patient with lung cancer, where that cancer is seen in that circle here. It's the opaque tissue in there shown in white. And what the group discovered about 20 years ago now is that when you compare cancer to normal tissue from the same tissue, there are a whole lot of epigenetic changes. You know, all these tags are disrupted. A lot of the histones are disrupted. There are really wholesale changes in the epigenome. And it took many years for people to believe it. And it took even more years for people to start taking it seriously. But we now know in models of cancer that we could actually cause cancer by reconfiguring these tags. I like to tell people that these are like bookmarks. And if we simply reconfigure the bookmarks, then there's a different instruction program. And we can see that in cancers, there's a very different instruction program when we study DNA methylation and histone modifications. So a few years ago, a group of us said, well, can we reverse that? Is there any way to put the bookmarks back to where they could be? And there were drugs that had been discovered many years before that were simply tools sitting in the laboratory to study. And we asked, if we give these drugs to people, will it help restore the epigenetic programs? And what we are trying to do in people is epigenetic reprogramming, the same thing that happens in embryos. And so this patient, who has lung cancer, was treated with one of these drugs. And what you see is after a couple months, that big circle showing the cancer shrank, and after about 6 months, the cancer actually went away.
Astonishing.
And we have hundreds of patients now where we see this phenomenon, where simply reshuffling the epigenome results in remarkable responses, and some of these last for years.
And so you've had treating, for example, cancers like MDS, the blood and bone marrow cancer?
So the first disease where this particular treatment was tried were forms of leukemias, and now there are 4 drugs that work on the epigenome that are FDA approved. Worldwide, there's more than 100,000 people who have received drugs in this form, and many of them have been helped. They prolong life, they work better than chemotherapy in these diseases. And there's a whole field of epigenetic therapy that has resulted from these studies, and virtually every large drug company now is trying to develop drugs that target the epigenome to do just that, reprogram genes and see whether that will help cure cancer.
So if I understand correctly from what you've said before, you're basically telling you're not going out to kill the cancer cells, exactly. You're going out to just give them different directions and tell them to be human again. Exactly.
So I've described this as this is different from the war on cancer. This is diplomacy in cancer, right? So what we're trying to do with these drugs is we're trying to change the instruction program of these cells to remind the cells that they should behave. You know, stop growing, you know, stop being poorly behaved cells.
Epigenetic change.
Yeah, exactly. I mean, many of us who are parents would like to do that to our teenagers from time to time, you know, but here we're trying to remind these cells that that they shouldn't behave that way. And in some cases, what we see is that the cancer doesn't go away, but it stops growing. You know, the cells have normalized essentially. In some cases, they start functioning normally. They start making the proteins that they should be doing, and we've really reversed the cancer program back to a normal program.
Fascinating. And Randy, we were talking about this before. You said that actually once you tell these cancer cells to stop goofing around and be human again, they actually die in the process.
Yeah, it's through a process of apoptosis.
Right. Apoptosis. There'll be an hour exam tomorrow, apoptosis,
right? A new word. It means programmed cell death, and in effect what they're doing is committing suicide. And so they cannot behave normally, but now the programs have been reset, as Dr. Issa has said, and it drives those cells basically, if you can't be normal, you must die. And that's what happens. The tumor disappears.
The implications are already evident in the giggles in the audience.
No,
if you can't be normal, you must die.
The implications are huge.
Homeostasis.
I mean, they're not only in cancer, they're potentially also important in, for example, treating or preventing neurological disorders. I mean, there are many places where the field of epigenetics is going to impinge upon our biology.
Neurological disorders. Francis, talk to us about the risks and the potential for using epigenetic directions to treat neurological disorders. Because that gets to mental health, it gets to all kinds of things in the brain. You've been working on some of this. Well,
yeah.
You have some warnings here, I believe.
Well, you know, we've taken, you know, for example, our rats that are getting high and low levels of licking grooming, we take them as adults, and if we put a drug in the brain that can reprogram these epigenetic marks, we can erase the kind of the effects of their early life maternal care. So we can improve the functioning of those individuals that did get low levels of care. But I think the key in those experiments,
the other way around too, right?
And the other way around.
Yeah.
So if we give them lots of methionine, we can increase those methyl groups and make them more stressed. But I think the key in all of these experiments is we're putting it right in the brain. If you're psychotic and I give you a drug that blocks your dopamine receptors, that might reduce your psychosis, but it'll also cause a lot of side effects, right? So it'll affect the way you move. Think of what these epigenetic drugs could do on a whole genome-wide scale across the body if we gave the drugs the same way that we give antipsychotics. It could possibly reprogram every cell in the body, and that's probably something we should be a little bit cautious about.
It reminds me of some of the geoengineering that is being talked about for man-made global warming that people want to do and say, well, yes, you can put sulfur in the air and it may cool things, but it'll do many other things to the complex system of the planet that we don't understand. So there's an enormous amount of caution, again, something we have to continually remember during this session here, that we have to be very careful.
But I'm going to ask you, Dr. Issa, about this. I mean, you're treating patients with these types of compounds. Are you finding problems with neurological disorders in the people that are being treated for cancer?
Right, so we're not, but keep in mind, Randy, that we are not. But keep in mind that the brain does not turn over. Therefore, the brain often does not actually see these drugs. The only cells that are seeing these drugs are the cells that are turning over, and cancers turn over more than other cells, and that's why we are seeing specific effects of these drugs on cancer cells rather than normal cells. But, I mean, that discussion is extremely relevant and important. We're only beginning to scratch the surface of how to do this. What is important about this patient and about the field in general is a proof of principle. We have shown that the epigenome is abnormal in cancer. We've had to convince many geneticists who thought cancer is all genetics and it can't be epigenetic. Now everybody understands that it probably is as much, if not more, epigenetic than genetic. And we've had to convince doctors that we could actually develop therapies based on this. And well, we have proof of principle. Many patients alive today, but who wouldn't be alive today but for these drugs. 10 years from now, we'll know a lot more about how to use them, when to use them, at what doses. We'll have many other drugs, and maybe we'll be able to find a way to change the epigenome only of nerve cells and not of other cells, or the epigenome only of breast cells in patients who are at risk for breast cancer. We don't know how to do that now, but the field is advancing so rapidly that it is not impossible to think that 10 years from now we could be doing that.
If I could alter the tone a bit and bring it down just a bit, financial considerations surely pressure what you all are able to do. And whenever we've been covering science stories before and we see real breakthroughs, we usually see groups who had financial stake in the earlier forms of cures that may now be found to be not the right form of cures. Are you finding any kind of denial from dealing with your work in medicine, from groups who were making money from the old kind of medicines that you're now trying to replace?
Well, I don't believe in conspiracy theories about, people who are blocking research. I don't think this is the case,
I wasn't
at least
suggesting that.
in medical research and in our fields. But it is true that the hardest thing to fund are the new ideas. Particularly, and that's where the National Institutes of Health is so important, because if you rely only on, say, drug companies to fund research, companies fund what is safe. They fund what they know. If someone comes up with a brand new idea like, wait a minute, cancer is epigenetic, not genetic, and I want to develop drugs for that, companies 20 years ago would say, you're completely nuts. Why would we put our money into that, right? So the considerations here isn't really active resistance, but issues of where the innovation comes from, and the innovation is what is hardest to fund.
Yeah, I'm going to go back to the fetal origins of adult disease susceptibility, which is what we started out with. Back when the epidemiological data were supporting this very strongly, many people did not believe this. Because scientists, you would think that this wouldn't happen, but it does. When there's no known mechanism for something to happen, they make the assumption it's the same thing as no mechanism. But there's one word different, which is known. And that's why our study that we originally had with the agouti mice was so important, because we now know the mechanism by which this occurs, at least in animals. So it gives credibility to all of these fields of epigenetic research. And one of the reasons why that curve that you initially saw went vertical after around 2004 is basically what it meant then is it brought in all of these groups in medicine under the big umbrella of epigenetic research, because a lot of people realized that indeed their research also needed to take into account epigenetic phenomena. Up until that point, and even to a certain degree even now, people and scientists are looking at biology through that I call genomic lenses or genomic glasses. I'm old and I have bifocals. In fact, I even have trifocals. We need to look at biology through both genomic and epigenetic lenses or glasses in order to truly understand how these diseases are formed, how to prevent them, and how to treat them.
So I'm hearing the sounds of a new paradigm. On more and more. Francis, have you run into any kind of denial as this brand new insight and way of seeing things has come along?
Well, we had a lot of trouble convincing people that maternal care could alter epigenetic changes. The most severe resistance I've ever seen in science. And I think, a lot of it's new. And and at the time, it was going against an existing dogma in science. And Dogmas in science can be useful. They give some guidance, some rules to the way things work, but they can also be very dangerous because they restrict the kind of questions we can ask. Right. And that's what we ran against, because there was this idea that modifications like DNA methylation, once they're in there and they can only be modified during early embryonic development, they're there and they can't be modified. So the fact that mothers could come along and modify the epigenome was something that was just a little bit too new at the time. Now everyone does it, it's fine.
In reading some of the research about your work, I came across this phrase, cryptic maternal effects. A powerful phrase.
Yes.
What are cryptic maternal effects?
Well, in this discussion of how traits are passed across generations, there's a large interest in these germline effects, that epigenetic factors are becoming incorporated into the germ cells and being inherited much like DNA. But there are a number of ways in which traits can be passed across generations, equally involving epigenetic changes, but in a very different way. And so we're looking at how mothers can shape generation after generation of offspring, not necessarily through changing the germline, but by changing the brain.
So not necessarily by changing the brain.
Yes. So if I change your brain, it changes the way you behave. If I change the way you behave to your children, it can change the way they develop. It'll change the way they interact with their children.
And this is sort of, yeah, of course, we've always known that, but the point is you guys are figuring out exactly how.
Mechanism. It's all about mechanism.
Mechanism. So it may be down from the DNA into the next germ and egg and down sperm and egg and the next sperm and egg, or it may be
leapfrogging
the moment of conception, but still passing from generation to generation. All right. So now let's, in our remaining 20 minutes or so, let's begin to open up some of the windows and take some risks in seeing what some of the implications of this extraordinarily exciting work you're doing are and what other parts of society it may affect. Randy, just before this started, we were chatting about the Civil War and epigenetics.
Now remember, this is a bit BS-y, right?
Yes.
I don't know anything about this.
But just to give you all a sense of new kinds of exciting conversations that now have at least the appearance of responsible conversation.
Right.
Give it
when we were sitting down, I made the comment that in the South, it is the, what would you call it, the stroke belt, the obesity belt, the cardiovascular belt, etc. And given what we know now about epigenetics and the fetal origins of adult disease susceptibility being potentially, at least potentially in humans, but in animal models surely involving epigenetic changes, that when you have severe lack of food, you can start setting up these types of situations that might be passed on from generation to generation. So one of the places in the South where you had tremendously a lack of food was during the Civil War. So it's possible that these diseases that we're seeing, these chronic diseases, might be increased in incidence. It might be a legacy of the Civil War itself. How that would actually be studied, I don't know. But I think it would be a fascinating study right here in our own country to look at that, if it would be possible through records to be able to look at that in a reasonable way. And I don't know how that would be done.
Right, and to be very clear, you're not saying this is the case, but this is now a reasonable question given what you've worked on and what they found
in the Swedish. It's an interesting postulate.
Right. Fair enough. Other aspects of the new wide open windows of legal responsibility that we may have here. Let's talk a little bit more about PTSD, the liability that we now understand PTSD has, because that has implications for the legality of war and for the inheritance from war and all of the genocidal and other regular horrors that go along with war. You're nodding again.
Yeah, no, we've been working with the Department of Defense to look at questions like this, and particularly how PTSD can affect parental relationships with children and how this might become inherited across generations. So trauma is likely going to be something that it has an intergenerational impact. And so we have to think about this when we're exposing individuals voluntarily to trauma.
And again, our understanding, coming back to some of the horrors of war, being that they leave the other population diminished in its ability to handle stress.
Exactly, and this is something that's going to affect the way they interact in an environment that's non-stressful. And I think that's one of the big issues with PTSD is that you're being exposed to a trauma that's just uncharacteristic of a typical environment. So how are you expected to then function within a normal environment?
And this again leads to the question of intergenerational equity and the joke I was sort of making. Are we looking towards a future in which it may not be unreasonable to think of suing your parents, your grandparents, or your great-grandparents because they didn't take care of the genome that you are?
Right. Or you could sue the government for creating an environment that's very bad for you.
Good luck with that.
Good luck with that. It's hard to sue governments, as we found out. But again, you're again pointing us towards a new sensibility among humans of what we are and what our governments are really responsible for. Again, this idea that we are each responsible for taking care of our own genome, not becoming alcoholic if we think we have a tendency towards it because of what it might do. By the way, what about alcoholism? Does that leave scars on DNA?
It leaves an epigenetic trace. It leaves it in the brain of the individuals who drink. It leaves a trace in the sperm of the individuals who are exposed to alcohol and their offspring. So very likely, and you think of the large-scale amount of alcohol that we consume, especially when we're younger, that this might affect reproduction, decades later.
You know that alcohol was actually tested in the agouti mouse model. 10% alcohol continuously in the maternal diet, and what it caused, surprisingly, was a positive adaptive response.
Uh-oh. Wait a minute. He's just talking about mice.
But we know
Drunk mice.
But we know, for example, that low levels drinking alcohol, for example, 1 to 2 glasses of wine a day, for example, help our cardiovascular system. But I always make the point that 1 or 2 glasses a day might be advantageous, in other words, positively adaptive. But I can almost guarantee that a gallon a day is not going to be positively adaptive. The point here is that a lot of these epigenetic changes and how the environment affects it is dose responsive. So at low doses you have no effect, then you get an optimal effect, and ultimately you get a negative effect. And most of the epigenetic effects and studies that have been done at a single dose, so you don't see this U-shaped curve. And I think a lot of the environmental insults that we're exposed to, in fact, with us with low doses of ionizing radiation, it's again positively adaptive up until you get up to higher, and then it's negative. So this is not an on-off switch, it's a rheostat, and it depends on where you are in that dose response curve as to what the effect is going to be.
And I want to make a comment about how much we don't know in this field, particularly about interventions that are dietary or alcohol or things like that. And sometimes what we think we know turns out to be completely wrong. For example, if you look at Randy's mouse model, giving the mothers extra folate looks like it's beneficial because it results in more of these little thin brown mice that have less cancer. But randomized studies in people suggest that if you give folate to older people, it may in some cases increase rates of cancer, it may increase mortality, and a lot of people are taking folate in their vitamin, which in my opinion they shouldn't be. So the point is that here's one situation where folate is good, another situation where folate is bad. Ultimately, we just don't know a lot about what really we should do to maintain an optimal epigenome, and I want to make sure people don't run into a pharmacy and start taking pills left and right.
Drinking a gallon of wine a day.
Thinking it will normalize the epigenome. I wish I knew what we should do.
Well, in fact, you're reminding me of what you might call fashions in the advertising noise.
Yeah, right.
For example, lately we've been hearing antioxidant, antioxidant, antioxidant.
Right.
But I'm hearing a few things lately from some of you and some of your colleagues that maybe antioxidants aren't always so great.
Correct. There's some evidence that in some cases they actually increase the risk of cancer.
And we showed with this agouti mouse system where low doses of radiation were positively adapted was completely negated by antioxidants.
Right.
So that's a negative effect of antioxidants.
Exactly.
Very interesting stuff.
The whole point being here that you now are showing evidence that you can begin to figure out about
exactly.
these things. Now, to open up some of the other wide windows here of where this is going, because I'm beginning to get overtones of a dreaded word from the early 1900s that made their way all the way into Nazi Germany, eugenics, meaning well-born. The eugenics movement, let's sterilize inferior humans was finally stamped out in America, but made its way into Hitler's Germany for horrendous, unspeakable results. And I'm hearing here again the possibility of this double-edged sword of human knowledge, like nuclear power when it came along, good and bad. There's a new potential here for knowledge about eugenics, is there not, that could be very frightening if am I wrong in having this concern?
Well, I think, Dr. Issa mentioned variability and the power of variability and how important it is for us to be different, right? We don't know what's optimal. We might think something is optimal now, but it might not be optimal 10 years from now. And so I think that's always the fear when we start selecting for traits based on genetic or even epigenetic factors, that yes, we'll do fine in the short term, but in the long term, and certainly intergenerationally, this might not be a very good move.
Well, I mean, even Francis, with your rats, I mean, those normal rats, the licker and groomers and the paws-off rats, right? Those animals were all part of the same variability of the population. So there's not something that's good or bad. I mean, if something dramatic is happening, you want an animal that's a little stressed because they're probably going to escape, whereas the other ones probably aren't going to escape. So it just depends on the environment, and the way the species survives is, and again, as you're saying, is to have this variability within the group.
A very basic important point here is that for survivability, it's good for the group, if it's going to survive, to have a variety of psychological types in it. It's been pointed out that Winston Churchill was chronically depressed like his father and grandfather, but it was a lucky thing at the time because chronically depressed people are organized by an external crisis, and so he could really lead them in the Battle of Britain against the bombs. So this wobbliness that you're talking about, this variability, is what's good if you watch it carefully and it's not too wobbly.
Right. Well, if it's too wobbly, it might have both good and bad effects. It is entirely possible that the same mechanisms that lead to, say, autism might also lead to Einstein. I mean, it might also lead to individuals who have extreme intelligence and therefore make great advances forward in the society. And if we stamp down those mechanisms and we say, okay, we're going to make sure that there's no variability and there's no risk of having these neurologic disorders, then we also stamp out, the emergence of the most creative people, which I'm sure if you looked at their epigenetics, they would be different from the norm. And so on paper, you would say we also should stamp these down because they are, they're not part of the norm. So diversity is what makes us evolve as a species, and diversity is what makes a society very successful in general. And so it's very important to be careful here not to, throw out the baby with the bathwater in that case. And we want some degree of wobbliness.
And to be cautious about putting negative connotations on various labels, such as autism.
Right.
It's differently enabled, and some Asperger's is said to be a kind of autism.
Well, exactly.
There's some great Asperger-y scientists and other people who brought us great things.
Exactly.
Definitely.
Probably
Particularly
more
in
of them in
science.
science than, in
Scientists always get a nice giggle going when I talk about Asperger's, because you all recognize something there.
Right.
We all know people with that in our field.
Right.
Who are brilliant.
Brilliant, yeah.
Absolutely brilliant, of course.
So-
And that, comes to around this diversity, right? And so there's really, humans are very diverse in their psychological makeup, in their brain makeup, in their abilities. And in some cases, these abilities, people who look off in society might be those who are going to save this society by discovering the next vaccine or, the next computer or something.
And you all now understand epigenetics to be a major part in what causes this variability.
Exactly, yeah.
And that's what's so exciting here. And it leads towards, we're getting towards the subject here of intelligence itself, which I was having a conversation with both James Watson and Eric Cantel, two Nobel Prize winners, just before this, and they were both talking about how intelligence is no simple G factor general, but it's a wildly diverse concept which has many different ways of being expressed. There's many kinds of intelligence and a myriad of mixtures of them. So again, this epigenetic variation sounds like it's more and more valuable for us to understand.
Right.
I've also heard it said that the supercomputer has not been invented yet that can even dream of beginning to map the epigenome the way we have the genome.
Right.
No,
Yeah.
I mean, you think the genome is complex. Think of all the different DNA methylation patterns you could possibly, all those histones, which can undergo many, many different modifications at various sites. And those are just two of many possible epigenetic ways of modulating genes.
So to be clear about this, these are just the first 2 or 3 with the acetylation that you all have discovered, and there may be dozens and dozens and dozens.
There certainly are.
Oh, there are.
There are
yes.
already known.
And they work in combinations.
Yes.
And so studying one may not be enough to understand things, and then studying 10 and then looking at how these combinations function is extremely difficult right now.
I mean, what we're doing in the field of epigenetics, I think, is that, again, coming from my computer science background and engineering background, is we're line by line decoding the programs of life. That's what we're trying to do now.
Say that again?
Line by line decoding the programs of life. And that's what we're trying to do right now. It is an immense thing to do. Immense.
Well, we're getting, and what I was saying at the beginning, jokingly, but wondering, are we beginning to look at a possible future where there may be pills that are going to no doubt be prescription pills that you could take to change your feelings of sexual orientation or your feelings of this kind of intelligence or that kind of intelligence or that's not impossible now.
So this is not impossible, but just keep in mind that our brains are mostly developed when we are in the womb and very early in infancy, and therefore the idea that a grown person is going to easily change their epigenome to change their psychological makeup, I think it may simply not be possible. It may be possible to influence the process in the womb and very early on, but there's a window where this might be possible, but past that window, that's going to be difficult. However, I do want to talk about something we haven't talked much about, is that this wobbliness, we keep talking about that. There's some uncertainty into where these method groups exactly are and how much there are. This is important for evolution, it's important for societies, but it has a detrimental aspect. And so this is the one thing that I alluded to, but I want to make sure I keep people in mind, is that this wobbliness exists within us. And it turns out that it changes as we age. And that might be one of the important societal things that we have to keep in mind in epigenetics, is that when we look at 70-year-olds or 80-year-olds, their epigenome actually has significantly drifted from when they were 20 or 10. And that wobbliness and that drift may be one of the contributing factors to many things that happen as we age, including cancer, but also perhaps neurodegeneration, perhaps heart disease, and other things. And that's the one thing where we have to be careful. We have to understand why our epigenome changes with age. How can we prevent these changes?
So you're saying that this may be able to lessen the effects of senility?
That's well, maybe,
Possibly.
and maybe lessen the chances of developing cancer, lessen the chances of developing
But it
heart
also
disease.
means, for example, lacing your flour with folic acid, which is going to be, it's been shown to be advantageous from reducing neural tube defects that later on in life, particularly when you're much older, could lead literally to an increased incidence of cancer. So what's good for one age group might not be good for the other age group.
And that's where taking care of our epigenome and taking responsibility for our epigenome might actually lead to longer, healthier lives. And maybe someday we'll find a pill that we can give to people that doesn't change your sexual orientation, but it does significantly change your risk of developing cancer or your risk of developing Alzheimer's disease or something like that. Genes are not strictly our destiny, and there might be ways to change that to some degree and to change the way we age.
You're nodding.
Yeah, that's what I think that's absolutely correct.
The possibility that we may now understand evolution itself and be able to take over control of evolution. Tell us what an imprinted gene is in about 30 seconds.
Well, to me, one of the most important things that has to be done, and this is from my perspective now, everybody here would have a different perspective, is that we have to define those genes in our genome that are called genomically imprinted genes. And they're genes that are expressed only from one copy, the one copy that's silenced epigenetically. The importance of this is that when you only have one functional copy, it's like you have a computer, but you haven't made a backup of it. So as a consequence, in this case, a single genetic event or a single epigenetic event can completely dysregulate the expression of the gene. These genes are developmental genes that are involved in growth regulation, and they vary greatly between species. And the information we have thus far from an evolutionary standpoint, that this evolved about 200 million years ago. And we still have these genes that are imprinted. But they vary greatly between species, suggesting that once the phenomena, the epigenetic phenomena of the mother and father turning off different genes, so you only have one functional copy, evolved, that software type approach was literally used to speciate.
So evolution discovered an even more...
A faster way that involves mutations and epigenetic events at the same time. It's almost a mosaic.
So we're running out of time, but that means that it looks like there's evidence that evolution itself discovered an even better way to speed up our faster evolution. And
the downside of this, though, is once you use software to evolve, you now become more susceptible potentially to diseases and neurological disorders, cancer, etc., because these epigenetic events that are required to silence the gene can be altered by evolutionary exposures.
And now we might be able to add a third stage of speeding it up because we have the knowledge to possibly help evolution itself. Well, listen, I want to get one question in here from the audience. I see a hand up going right there. Yes, ma'am? No speeches, please. Keep it short and quick. Something we need to know.
Are you saying that this can revert with metastasized cancers? And if so, how far along in the metastasization have you actually seen it revert?
The question had to do whether this therapy could reverse the metastasis process, and the answer is yes. In those patients who are benefiting from this, it's not just the primary cancer that's resolving, but also multiple metastatic lesions in some cases, and we're hoping this could affect cancer survival by affecting many different metastases.
Okay, answered. It's a beginning. Another yes.
To what extent does the bacteria, the metabolome in the body affect methylation or acetylation?
The question is, to what extent does bacteria in the body affect methylation?
It's a great question. I believe it does. We have a little bit of data that it does, but it's almost completely unknown right now. But I think that 10 years from now, we will have discovered that the bacteria in our body are essential for maintaining the epigenomic health of the cells that are exposed to them, such as our GI tract.
Is there a question in the balcony?
I have a question for Francis about maternal care. Does that apply to maternal or caregivers?
Is it just for maternal care or any caregivers?
Right. So our rats are all focused on maternal care because the fathers don't participate in care in any way, shape, or form.
Among the rats.
But when you have a species where the fathers do participate in the care, they can have similar epigenetic effects on offspring. And there's more and more research being done on species in which there's exclusive paternal care or caregiving by other individuals. And they have very similar effects. So it's really about your experience as an individual with whoever is providing the care, rather than directed by some specific individual, whether it's mother, father, or
Francis,
someone
can't
else.
you even substitute a
Well, yeah.
paintbrush?
So in rats, you can use a paintbrush and actually get the same effect.
Instead of licking.
Yes,
Yeah.
instead of licking. But I don't recommend it as a child rearing strategy.
But you can also have other mothers raise the pups and have exactly the same effect.
Yeah.
So caregivers would have the same effect as the mothers, even if they are not mothers yet.
Exactly.
I wonder how many babies now are going to be tickled by paintbrushes. Yes.
Have you done any expression arrays with goody mice?
Say it again.
Have you done expression arrays?
Repeat the question so that we can
Have you
understand
done
it.
expression arrays with mice, the goody mice, to see whether or not, some nutritional exposure, whatever, alters the...
Jargon alert. Expression arrays? What's that?
They're small plates or whatever that look at the expression of a whole bunch of genes at one time. And so you could look then to determine whether or not a certain exposure altered the expression of a bunch of different genes and what they are. The simple answer to that, we have not done that, but it could be done. We've only looked at 2 genes in the Goody mouse, and both of those genes we looked at were changed in the same way. But that doesn't mean that there wouldn't be other genes that would be reduced or whatever, but we just have not looked.
So one more question. Obviously what that tells us, for those of us that didn't understand that question,
Or that answer.
or that answer, is, I did get a sense though that you're just at the beginning.
We're at the
You have a lot of
beginning of
stuff to do. All right,
That's it.
one more. I see a hand up there. Don't have any idea. It seems to be a human holding a hand up. Yes.
Any thoughts about the relationship between culture and epigenetics?
Between culture and epigenomics. A great last question for each of our panelists. Randy, any epigenomics and culture? We can come back to you.
Let me think about this.
All right,
Okay.
Francis?
So culture is another exciting route through which you can have a transmission of traits over generations or characteristics of the general population. So it is likely, often geneticists will refer to that as a kind of a soft way of transmitting traits. And certainly it's likely going to recruit epigenetic changes within whole populations to foster changes across generations. So I think culture, epigenetics, parental effects, and genetics really form kind of a collective of ways to transmit traits across generations.
And culture affects what we eat, it affects
Right.
how we eat, it affects what we drink, and ultimately these things on the long run affects how our epigenome ages and therefore our risk for age-related diseases. One of the most remarkable observations in cancer research is if you take migrants, people who move from one country to the other, within one generation, they adopt the cancer risk of the country they move to. So
Wow.
Japanese people, for example, had very low colon cancer rates in the past. When they moved to the US, they have the same colon cancer rates as the US, and that's partly culture. And so it is very important, and it's partly through the epigenome.
Randy?
My only addition to that is really, I mean, that was a wonderful answer, is that, from our original study, what we showed is very relatively subtle changes in nutrition very early in development can have profound effects on ultimately disease susceptibility through altering of the epigenome. So to the degree that culture is altering even the food that you're taking in, et cetera, you can see very clearly if we have the same phenomena occurring in humans, how culture could have a dramatic effect on the epigenome and also diseases.
Whole new world to study out there. Great last question. So let's give them a big round of applause.